Okay. Hi. So thank you very much, and thanks for the organizers for putting up this nice meeting. Thanks, Prabal, Ambarish, and Tanveer. I'm going to talk about self-assembly of filaments. So I would talk about self-assembly and 3D organization, a little bit of dynamics of some of those tracks, and I would present some physical models thinking about these systems. So like this meeting, in our lab, we work on many diverse areas. We work on chromatin, we work on microtubules and cytoskeletal filaments. We also work on aggregation of proteins. So I'll try to talk these first two topics mainly, and a little bit, if there is time, about this. So the first part is nucleosome positioning and chromatin assembly. As you all know, we have many different cells, like a skin cell, for example, or cells in our brain. All of these cells have exactly the same DNA, but these cells behave very differently. The cells in our skin behave very differently from the skins in our eye, or cells in our eye, or cells in other parts of the body. So how do they show this very different behavior, even though they have exactly the same DNA? And the answer is, the DNA is organized in these cells differently, and therefore the DNA organization is very important beyond the sequence of the DNA itself. And one of the very interesting questions is, how is chromatin organized in vivo? And this is a cartoon of chromatin organization. There is a lot of details. I would just want to say now there's a lot of details on the organization of the chromatin itself. And in different cells, in different situations, the chromatin will be in different state in the thermodynamics of statistical physics language. They'll be in different macro state or micro states, and therefore they will function differently. And the state of the chromatin is actively maintained. It's not an equilibrium state. They're actively maintained by ATP-dependent chromatin remodeling machine. This is important to understand, that you have to constantly supply energy to maintain the chromatin in the state that you would want. The different levels, so in textbooks, if you look at all the textbooks, for example, Albert's the famous textbook, it would describe different levels of organization of chromatin. For example, the DNA, which is the first level, which is the sequence, like one-dimensional sequence. And the DNA is then packaged, wrapped around this yellow ball-like histones to form this first level of packaging. So these are called nucleosomes. So this is thought to be like an array of nucleosomes. And then it is imagined that it is further packed in a hierarchical manner to finally you have, in the mitotic phase, you have mitotic chromosome. So in the interface, there is some higher order organization is what is being taught. And even though there are various cartoons available in the literature and in the textbook, none of these cartoons have been experimentally proved or verified. So we do not know how exactly is chromatin assembled beyond this level. So what we know is that this level we understand very well, but beyond this we do not know how exactly chromatin is organized. It's still being the ongoing research in the field. So, but even in this level, the first level of packaging where you have DNA and on which you have nucleosomes, the interesting question is, how are nucleosomes organized along the DNA? Are they organized randomly, or is there a particular pattern for the organization of the nucleosomes? So this is a question that is being discussed last one decade or so, discussed a lot and studied a lot. And the answer is, of course, it's not randomly organized. There is a very specific organization, at least in some specific locations. For example, what is plotted here is the nucleosome organization along the DNA near transcription start site. So this point, somewhere here, wherever there is this is a transcription start site, and this is the typical coding region. And this is the nucleosome organization near transcription start site. This y-axis is nucleosome occupancy, which is basically the probability of finding nucleosomes at that region. And as you can see, and this is averaged over all genes in yeast. So if this was completely random organization, you would expect a flat curve. but there is a very nice pattern indicating that the organization is not random. There is more probability to find nucleosomes at particular points as opposed to some other points. So interesting thing to note is that near transcription start site, there is this nice pattern. However, this pattern is maintained only if there is ATPs available. For example, this Frank Pugh lab did nice experiment of nucleosome positioning in vitro with cell extract with ATP and cell extract without ATP. And what they found is that if you have cell extract with no ATP, you do not get this nice organization. You get almost a flat curve. And so this is interestingly shows that ATP and ATP-dependent molecular machines are necessary to obtain, to maintain this particular pattern of nucleosome positioning. It is also now understood that stochasticity plays an important role in nucleosome organization. For example, this is an experiment by Small et al. And what they did was, and similar experiment is also done from the Boyger lab. What they did is they took yeast exactly under the same condition, many yeast cells, and they look at the nucleosome positioning in the 4-5 promoter, and all yeast cells are exactly under the same condition, but when you look at the nucleosome positioning in the promoters, you would get different populations having different positioning. So there is, even though the cells are exactly under the same condition, the nucleosome organization is slightly different, which would imply that there is stochasticity plays some role in the nucleosome organization in promoters and in other regions. So this is also very interesting for a physicist. And based on this, we thought about this and did some work where we asked some questions. Given that nucleosomes are dynamic, which is also known that nucleosomes can be nucleosomes slide with the help of molecular machines, and nucleosomes get disassembled. So given that nucleosomes are dynamic, they can bind, dissociate, and slide. What must be the underlying dynamical rules to obtain the experimentally known occupancy pattern? So we experimentally know some pattern. What should be the dynamics behind it so that one would end up with this pattern? So this is one question that we asked. We also asked, what is the role of ATP-dependent remodeling in maintaining this experimentally observed organization? If you want to maintain that organization away from thermal equilibrium, what must be the dynamical moves that must be going on? How nucleosome organization would influence the three-dimensional looping of chromatin, and how certain known histone proteins might affect the chromatin organization. So these are the questions that we started asking. So this is one work we did regarding the nucleosome. There is a slide which is kind of missing in it. Oh, okay. There is a slide probably missing. I don't know why it is showing black when I go there. And it's not seen in my computer, so I don't know what happened. But basically, we have a model where we have a one-dimensional track on which particles bind, dissociate, and slide. And these rates we model in a sequence-dependent manner, and we put ATPase activity into these rates, and we simulate using Monte Carlo simulations. And this is one of the results. For example, one of my students, Josna Parma, she did a lot of this research. And for example, when we look at occupancy with and without ATPase activity in our simulations, we get similar to what is seen in experiments. I will explain this in detail if any of you would want. Okay, we also studied, for example, the stochastic organization in the promoters. So there are 8 different states. For example, this is a state in the promoter, all the 3 nucleosomes are present. There is another state where are only two nucleosomes present, but that can be in 3 different ways. Another 3 states, only 1 nucleosome present in 3 different ways. And there is a state where there is no nucleosome. So there are 8 coarse-grained discrete states of nucleosome assembly organization in the promoters, and they have different probabilities measured in experiments. And our computation can predict what are the ingredients necessary to obtain this kind of organization. So that is some work that we have been doing. I'll mainly talk about the second part of the chromatin organization, which is how do nucleosomes influence the 3-dimensional organization of the chromatin. So as you can see, nucleosomes play some important role in 3D organization, and there is a lot of details which I wouldn't go into, like there are certain marks like modifications, post-translation modifications, and so on and so forth. let us think about what is known in the textbooks. So if you, again, look at textbooks, what is taught about higher order organization of chromatin is models like zigzag and solenoid models. What do they say? They say that chromatin is assembled in a regular manner, into a regular helix-like manner. And most popular model is a zigzag model, where there is a zigzag-like pattern of nucleosome arrangement, and the chromatin is thought to be arranged like this by some of the earlier experiments indicated it, and some of the prevalent models suggest that this is the organization of the chromatin. And this is also called a 30 nanometer fiber of chromatin, where the width here is 30 nanometer. Nucleosome has about 10 nanometer width. So 2 nucleosomes, 20 nanometer, and some linker DNA in between. This is about 30 nanometer. So it is being argued that, or it is being thought that chromatin is organized in this 30 nanometer fiber in this fashion. This is one of the higher order organization of the chromatin, is what is being told in the textbooks. A lot of in vitro experiments also supported this. For example, this is a in vitro experiment by Richmond lab, where they did 4-mer crystal structure. So they have 4 nucleosomes, and they looked at the crystal X-ray crystallography they did, and they looked at the arrangement, and they again got somewhat like zigzag. They also did in vitro electron micrograph of 46-mer, showed zigzag. But there is a lot of caveats in these experiments. And for example, Mark, when we had discussion, there's a lot of caveats gone behind obtaining this kind of patterns. And nonetheless, this is what they got. They got zigzag, and this kind of reinforced the notion that chromatin is organized like a zigzag structure. Let us quickly look at what is the physical principle behind this zigzag model. So the prevalent theory in the literature is the zigzag organization of chromatin. And if you look at what is the physical ingredients gone into this model, there are 2 factors that decides this organization. First factor is the fact that when DNA enters and exits, so DNA is wrapped in nearly 2 turns. So if you imagine this as a small vector entering into the nucleosome, and this is as a small vector exiting the nucleosome, these 2 vectors make a specific angle. So there is an angle of entry and exit nucleosomes. For example, there is an angle called alpha. So some specific angle, nucleosomes, in general, any protein binding to DNA would bend the DNA into some specific angles. So that is the first physical idea that is used in this models. And the second idea is that the linker length is relatively short compared to the persistence length of the DNA. So the linker length is 20 to 60 base pair. Typically in yeast is on average 20, and it could be 60 or 70 base pair. So which is much smaller than 150 base pair, which is the persistence length of DNA. So since this linker length is very small compared to the persistence length, this region will be nearly straight. So there is this angle, and there is a straight, stiff linker region. And these are the main 2 ingredients. There are other ingredients, but with this entry-exit angle of DNA and the stiffness of the linker region, one could simply argue for the physical, you can make a physical argument to propose a zigzag model. So this is proposed by Woodcock et al. In 1993. And it turns out that many of the recent experiments in the last few years, there is a flurry of experiments. All these experiments, they try to probe chromatin close to in vivo or in vivo. And what they found, they could not find any regular order. They could not find the 30 nanometer structure, which is supposed to be there. So, for example, these are some titles. Analysis of cryo-electron microscopy images does not support the existence of 30 nanometer chromatin fibers in mitotic chromosome in situ. Human mitotic chromosomes consist of predominantly irregularly folded nucleosome fibers without a 30 nanometer structure. There are many other experiments. I just showed 2 samples, and many labs studied this. And with recent emergence of Hi-C and chromatin conformation capture experiments, even they argue that there is no clear regular structure of chromatin, and therefore chromatin is irregular in vivo, is what is all the experiments indicate. That is a puzzle, that why, if in vitro experiments and models suggest this kind of regular 30 nanometer structure, while experiments do not see in vivo. So we, in this point where we started thinking about it, and when we looked at all the models so far only consider histones, core histones plus H1. Some models also consider H1, and that too very regularly arranged histones, like they are organized in a regular, very nice manner. And what we thought is that no model so far has accounted for the role of DNA bending proteins. So known histone proteins like NHP6 in yeast or HMG in, these are some examples. There are many proteins that bend DNA, including many of the, including chatter binding protein, and many proteins bind to the DNA and bend DNA. So what would be the chromatin organization in the length of a gene or a few genes, which is of the order of a few nucleosomes, given a concentration of DNA binding proteins and a given nucleosome organization? So this is the question that we started thinking about. Given a nucleosome organization and a concentration of DNA binding proteins of particular kind, what would be the chromatin organization? Can we look at it? So this is what we tried to simulate. We did two kinds of simulations. So Gaurav is a student who did this work. So we did Brownian dynamic simulations with some details, and we did polymer models, which is called a freely rotating chain model for polymers, where each polymer is created by a set of 3D vectors, and wherever there is a nucleosome, there will be a specific angle, theta n, which is the nucleosome angle. Wherever there is a protein, there will be a specific angle called theta p, which is the protein angle. So we could make a freely rotating chain-like models. This is one thing we did, and to support this, we also did a little bit more detailed Brownian dynamic simulations, where imagining DNA as a bead spring chain, nucleosomes, two ways we took for a simpler nucleosome which makes an angle, and H1-like proteins which would hold this angle to specific numbers. And there is extra known histone proteins like red here, which would make some specific angle, which is like this theta p, some specific angle they will make on the linker region. So with these ingredients we simulated, and the results are the following. So if you do not have this non-nucleosomal protein, one would get nice regular zigzag-like pattern, even though some is not very, there is a nice pattern, and we'll quantify the shape of this. And with this non-histone proteins, of course, you get an irregular structure given some density of non-histone protein, which we will discuss. Again, this is the Brownian dynamic simulation, and this is the freely rotating chain polymer models. And even this, if you do not include that non-nucleosomal proteins, you would get regular structures. And if you include non-nucleosomal proteins, we will get irregular structure. So this is what came out of our simulation. So the question is, what is the critical density of this non-nucleosomal proteins beyond which the zigzag would disappear? Because if you do not have any non-nucleosomal protein, you will get nice zigzag structure. And if you add a lot of these proteins, zigzag will go away. So is there a density beyond which the zigzag will go away? Is that density biologically relevant? Because otherwise, this wouldn't make sense. So what we computed to quantify this structure is probability of neighboring nucleosomes to interact, because this is something that a lot of people calculate in experiments. So for example, if you look at a zigzag structure, ith nucleosomes and I plus second nucleosomes, that is next neighbor, are nearby in 3D space. So this is the second neighbor, that is next neighbor. Here in this picture, the neighbors themselves are near in 3D space when there is a bending of DNA. So this I of k, what is plotted in the y-axis, is the probability that the kth neighbors are nearby. Okay, so that is what is I of k. So if you plot I of k versus k, where k is the kth neighbor, for a zigzag structure, which is the blue, the peak would be at k equal to 2, which means is nice zigzag-like arrangement where the peak is at 2. As you add other the proteins that bind onto the linker region and bend the DNA, this peak would come down. And as you add more and more, the peak would further come down and come down. And there is a point where these two, the peak at k equal to 1 and the I of k, that is I of 1 and I of 2 will be equal. And it turns out that the known histone protein density is around 30 to 50%, depending on the precise nature of this protein. The protein would bend with certain angle. Depending on the bending angle of the protein, typically 30 to 50% of linker regions are bound. Then one would get this I of 1 is equal to I of 2. So that is neighbor and next neighbor nucleosomes interact. So the neighbor interact and next neighbor interact with equal probability. And this result, interestingly, is comparable with recent micro-C, which is some kind of a high C done in yeast, where they could look at nucleosome level resolution high C, and where they suggest, for the technical, they use micrococcal nuclease to digest so they could get high resolution. And this is from Rando Lab. And they also say that this I of 1 is roughly equal to I of 2. So at least this is the parameter regime where I of 1 will be equal to I of 2. That is when known histone protein density is about 30 to 50%. Correct. If the distance, if the cutoff, we put a cutoff, and then cutoff is the nucleosome distance plus tail distance. If that is below that, we call it interacting. Yes, nucleosome, yes. So how many linker regions are bound by this? So you have many linker, if between two nucleosomes there is a linker region, which linker regions are bound and which are not bound? Yeah, number of, in the model, yes, in the model, yeah. So there are two parameters mainly. So one is the density of this known histone proteins bound on the DNA, and there is a linker length, which is another parameter. And these two parameters, if you vary, you will have a nice phase diagram like plot, where small linker and small protein density, you would get a regular structure. And if you have large linker length and large protein density, you would get more irregular like structure. There is another parameter, which is the bending angle of the protein. Some protein could bend only very small. So you need a good enough bending angle. So if a bending angle is between 90 to 140, then this is the corresponding protein density which is required to get irregular structure, at least in our models. Of course, so this is the parameter regime that, according to our model, would give one this kind of irregular structure. So that is the first part, and it's nicely 30 minutes. Okay. The summary of the first part is that physical models for nucleosome positioning, where there's something we study. We study dynamics of nucleosomes and how nucleosome positioning affect 3D organization of chromatin. And we also look at DNA bending known histone proteins. How will they influence the three-dimensional organization of chromatin? So this is the first part. Okay, why is it? The second part is the other track which all of you are interested, which is about microtubule. So we also study a little bit about microtubule dynamics. So we look at models for microtubule dynamics where we combine the chemical kinetics with the filament mechanics. So this is something about that we will discuss. So there are many theories for microtubule dynamics. So there have been many theoretical and computational studies trying to understand microtubule dynamics, and each theory is successful in some aspect, explaining some aspects of microtubule dynamics. But there's no single theory that explains everything. There's many phenomena that is experimentally measured in vitro and in vivo. And to quantitatively explain all the phenomena by one theory is like very difficult. And if we accept some of the existing theoretical notions in the field, it will lead to some kind of paradoxes. So we'll see what paradoxes will emerge if we accept some of the theories. And our aim is to build some consistent theory that would explain. The hope is that to all phenomena, we are going to start there. So let us see what we are thinking about. So what we know is that GTP-bound tubulin and these protofilaments bound to GTP-bound tubulin protofilaments are relatively straight. So this is what the textbook models again tell us. So this is a straight GTP-bound protofilament. So the cap would make this protofilament straight. When this GTP hydrolyzes to GDP, this protofilaments would start curling. So there is a bend, and the protofilaments, the GTP-bound tubulin will curve, and therefore this will be in a curved state, this is the EM picture of this. And this is something that we know. We also know that in the microtubule cylinder, the lateral bonds between protofilaments, so the lateral interaction between protofilaments, they hold the microtubule in a stable state. So the lateral bonds are important. The bonds between protofilaments, which are called the lateral bonds, they are important to hold them in the state. We also know that the GDP protofilaments would want to curve out. So if everything is hydrolyzed, this would want to curve out, destabilizing the microtubules. So if you think of a microtubule where all monomers are hydrolyzed, all dimers are hydrolyzed, the power struggle between inter-protofilament interaction and the bending elasticity is what decides the stability of microtubule. And the notion, the popular notion in the literature is that when everything is hydrolyzed, the dominance of bending energy in the GDP state leads to unzippering and shrinkage. Since the bending energy is much larger than the lateral interaction energy, that would force the microtubule to curve out, and that would lead to unzipping of these microtubules. We question this notion through some calculations. So another thing we know is that typically experiments measure 4 parameters, very coarse-grained parameters for microtubule, which is the growth velocity, which is the slope of this curve, the shrinkage velocity, which is the slope of the shrinking part. There is a catastrophe frequency, which is how often the catastrophe happens, and there is a rescue frequency, which is how often this goes back. At least in vitro experiments measure this, and in vivo there are various other things, but these are the typical 4 parameters that experimentalists measure. And the question is, what are the microscopic origins of these parameters? How do these parameters emerge? Because there is chemical kinetics, which is polymerization, depolymerization, and there is also some kind of filament elasticity which would curve out the protofilaments. So some combination of chemical kinetics and the filament elasticity together should decide at least some of these parameters. So and all of these processes are not like single-step processes. There are multi-steps, and therefore, how do they decide some of these parameters is an interesting question. No simulation or theory could reproduce, for example, the observed shrinkage value of shrinkage velocity. So microtubule has a particular shrinkage velocity. In vitro, it's about 30 micron per minute is a typical in vitro shrinkage velocity of microtubule. From putting in this chemical kinetic details, and either you could take this as a parameter, or if you put in all the details of the bending elasticity and so on and so forth, can one get this number of 13 micron per minute? So nobody has done that. So simultaneously, so this plus the length of this ram's horns, this is sometimes called ram's horns, it's like a horn of ram. So what is the typical length of this is estimated in experiments. So this is another interesting experimental data that we have. So we have a shrinkage velocity and the mean length of this ram's horns. So these two together is not explained by any theory. So we started we decided to start from there. That can we explain the shrinkage velocity as well as the average length of the curved regions? So our attempt is to understand how mechanics combined with chemical kinetics, chemical interactions determine one of these parameters, one of these 4 parameters, which is the shrinkage velocity parameter. That is the one we would focus today. This work was done by Ishutesh Jain, who is now a postdoc with Fong Chang and Curie. So the question we asked is, how fast will a GDP microtubule protofilament unzipper? So this is the first question we asked. So unzippering is this curving out of a phenomena. So what is the speed of this? In other words, what is the time scale of peeling off? This is like peeling off. So what is the time scale of peeling off of these protofilaments? How does the competition between inter-protofilament interactions and the bending elasticity influence the unzippering velocity and the shrinkage rate? So there is a velocity of unzippering and there is a velocity of shrinkage. How will this be determined by this competition between this inter-protofilament interactions and the bending elasticity? Do thermal forces play any role at all? Or is it just the dominance of the bending energy dictate everything? Or is there a entropy or thermal forces, do they play any role? And if you think of shrinkage, there is a filament can shrink in 2 ways. They can all unzipper, and that would destabilize filament and you would not see any filament under a microscope. So filament can just shrink just by unzippering in principle. There is also depolymerization. So how does it work? What is the order? How does it happen? Right? How does this combine? How does this together work? And the essential features needed to reproduce this L versus time curve. So you have length versus time curve is typically that you would explain, that you would see in experiments. So what are the essential features needed to reproduce this plus catastrophe frequency plus Ram's-Horn's length and all together? So comprehensively, if you want to explain various things seen in experiments, what are the things needed to be put in to a model? This is the question that a physicist is interested to think about. So we started with a little bit detailed model where we wanted to consider 3-dimensional nature of the cylindrical nature of microtubule. Of course, microtubule is 13 protofilaments, and simulating all the 13 is like very difficult, computationally expensive. So we started with 3 protofilaments, because 3 is the minimum number that you can put in to make a cylindrical shape. So if you have 3 protofilaments and they all interact with each other, so the interaction energy is, so each of these protofilaments interact with this neighbor through some kind of interaction, so harmonic potential we put in, which has a cutoff. So this is when the distance between 2 neighboring protofilament monomers is Li. If Li is less than some Lmax, it is like harmonic. And if Li is above Lmax, it is a constant, which is Lmax minus L0 square. So this is a potential which is like somewhat like this. So a harmonic up to a point, and beyond that's a constant. And each of this protofilament has a bending elasticity given by typical worm-like chain-like model, where there's a bending stiffness and there is a intrinsic angle 1 minus cos theta I minus theta 0, where theta 0 is 0 if the ith protofilament is a ATP-bound protofilament. If the ith protofilament is a GDP-bound, if ith dimer is a GTP-bound dimer, then theta 0 is 0. If the ith dimer is a GDP-bound dimer, then there will be a specific angle theta 0, which is known from the structure of, which is estimated by various experiments. So there is a bending elasticity with an intrinsic angle, preferred angle for GDP-bound protofilaments. So we took this. I don't know why. We also have, of course, a one-dimensional, much easier model where we can get some analytical insights, where you have just a stiff protofilament, which is fixed here, and you have a one filament, which is a semi-flexible polymer, exactly with the same interactions. So there is a spring or a harmonic spring, which is like a bond which has a cutoff. So this is a harmonic bond between this bottom line and the protofilament, which was like a lateral interaction. So this would mimic a lateral interaction. And this one is the protofilament of our interest, which has, if everything is GDP, this would like to have an intrinsic curvature. This would like to be curved just by the same expression, 1 minus cos theta I minus theta 0. So the parameter that we measure here is this Rx. Rx is basically the distance from everything straight conformation. This distance is called Rx. If the protofilament is completely unzippered, the Rx will be very large. If Rx is 0, means protofilament is straight. So Rx is like an order parameter that one would keep track of in the simulation. So we did Monte Carlo simulations for this, yes. So it is length with some cos, some angle parameter circle, because there will be a circle at the end of it. So there will be some sine, cosine, this plus, close to length, but with some correction of given by the geometry. Because it can go like this, no? So it can do like this in principle. So up to this, with some correction given by this geometric factor. So we compute the free energy. Now, how do we calculate free energy? Of course, from Monte Carlo simulation, we can calculate P of Rx, the probability to have a particular value of Rx. This is something that we can calculate in simulation, and log of probability is like a free energy. So we calculate log of Prx, which is the free energy of this unzippering tip as a function of Rx. So if we plot this free energy, F of Rx, which is log of this P of Rx, what we get, this is what we get. So these curves are for different value of delta E. Now what is delta E? Delta E is the maximum spring energy minus maximum bending energy. Maximum spring energy is this value. So this minus is essentially the measure of spring energy minus the bending energy, the energy to completely straighten a curved dimer from its intrinsic curvature. So this is the parameter. This is essentially some the lateral energy parameter and the bending energy parameter. So this is lateral energy parameter minus the bending energy parameter. This difference is delta E. So if delta E is so this curve, if you take the red curve, delta E is 3. Delta E is 3 means the lateral energy, the spring energy, is much larger than the bending energy. So if the lateral energy is much larger than the bending energy, the minimum of the free energy will be when Rx equals 0, which means that the protofilaments are nicely arranged and the microtubule is stable. The other extreme, where EB is much larger than ES, that is the bending energy is much larger than the spring or the lateral interaction energy, that would give this other extreme where the minimum is somewhere here, where Rx is, I have only brought it up to this will go up to the length, close to the length of the microtubule itself. So the minimum is somewhere here, where the Rx is a very large value, indicating that the microtubule is unzippered. Now, look, the x-axis is Rx. So you have a free energy landscape, and unzippering is equivalent of moving in this free energy landscape. So I can ask the question, if you have a particle in this free energy landscape, how fast will it move? How long it will reach from point A to point B? And what is the time scale? And so on and so forth we can ask. And somewhere here, interestingly, both compete. For example, and this is a little bit more zoomed in version, where here, this is the free energy landscape. Interestingly, this is a multiple minima, and we have some analytical thought on why there has to be some multiple filament, at least in the single filament picture, which I would explain after the talk if anybody is interested. So this is the 3 extreme, where this is the bending energy dominated, this is the lateral energy dominated free energy landscape, and here the parameters of bending energy and lateral energy will compete with each other. Now we ask the following question: if this microtubule is to unzipper in this landscape, how long it will take to unzipper? What we know experimentally is that since the shrinkage velocity is half a micron per second, that is 30 micron per minute. This is an experimentally given to us that shrinkage velocity of microtubule cannot be faster than half micron per minute. Therefore, you do not want the unzippering to be faster than that. So unzippering has to be slower than half micron per minute. So we ask the question, which of this landscape will give us the unzippering speed, which is smaller than half microns per minute? So interestingly, we did not get this. We got somewhere close to this, where both this energy compete with each other. Yes. Yes. So the tubulin dimer is like 8 nanometer. So, and this, if I have a tubulin and this curves like this, that is about the first minima. So 120 nanometer is the typical intrinsic curvature, would lead to a curvature of 120 nanometer. That is what Rx is, 120 nanometer. So this, yeah. Yes. Yes, so this number, we have number of, this is 8 nanometer, yes. 8 nanometer is the length of the monomer. So we have n monomers. Each monomer is 8 nanometer. That would set the length scale. Yeah. We have done it. So right now, no. In the work we have added depolymerization to this, which maybe I don't have it because of the lack of time, but I will tell you. In the work we published, we also have depolymerization. But this itself tells us that the landscape cannot be this. So this is what people would have imagined naively, that the landscape is this, where the particle, if you leave here, everything is GDP, so it should just shrink. If I put a particle here, it will shrink much, yeah. With depolymerization also we have done exactly the same result. Yeah. Yeah. So with depolymerization, I'll show you the result. So this is the velocity versus this delta E. So when delta E is of the order of kBt, few kBt, is where one would get the velocity of same order of magnitude as that of the experimentally seen velocity. That would question the notion that delta E is much smaller than zero. Delta E is much smaller than zero is the notion. And as Anatoly said, we also did exactly the same thing with depolymerization and all details. And because of the lack of time, I haven't put it here, but I'll show you just after the talk. Yes. Now, what we are only comparing is the shrinkage velocity, where a completely hydrolyzed microtubule in vitro will shrink with a velocity of half a micron per minute, which is some kind of a constraint for us, because the shrinkage shouldn't be faster than that. Because if the unzippering is faster than half micron per second, then within a second it will just unzipper, right? You don't want that. You want this unzippering to be slower than half a micron per second, because that is what the velocity seen in experiments. So that is a constraint for us. And we ask the reverse question: if this has to be, unzippering velocity has to be smaller than the shrinkage velocity, how should the landscape look like? So to summarize, we have a model with mechanics plus kinetics, which I haven't shown here, which is the depolymerization rate, which can obtain experimentally sensible unzippering and shrinkage velocity. We can obtain, so we also calculated many other quantities, like the length of the curved region, and we get sensible results, like the average length of the curved region is something known in experiment. And when this depolymerization happen in the landscape that we discuss, the average length of the ram's horns is comparable to experiments, which is of the same order of magnitude, and the length versus time data also with no ad hoc assumptions. Yeah. Per monomer. Yes. So then, yeah, it's whatever because microtubule, the velocity is independent of length. So we can only say per monomer. The shrinkage velocity is independent of length, right? So then one would expect a length dependence in the bending velocity, which is not seen. Okay, so yeah, this is our microtubule model. I'll just show a couple of slides on other things that we developed similar ideas to study protein aggregation as well, because of late protein aggregation has been a very interesting question in many different fields. Like, of course, disease-related, amyloid-related protein aggregation has been an interesting area for many years now. Many other factors, many regulatory factors like transcription factors and many other factors aggregate, and that helps in the regulation itself. So one of our students, with my colleague Samir Maji, we started thinking about aggregation of protein, first in the context of amyloid aggregation. And the question that we asked is, if you have a diverse set of proteins, independent of their sequence, they all aggregate into diverse and form diverse kind of aggregates. That is, sequence exactly doesn't matter. Very many different sequences aggregate to different structures. So that means there has to be some underlying physical mechanism behind this. So we wanted to think about, again, from a physicist perspective, starting from simple models, how the assembly can be examined. For example, to assemble into order structures, and there is diverse set of order structures. And we again simulated peptides, small polymers with two parameters. One is the bending stiffness of the peptides and the interaction strength, so which is like a Lennard-Jones potential. So we have just bending stiffness, that is one parameter, which is k into 1 minus cos theta, and there is a Lennard-Jones attraction. Only these two interactions, and we studied peptides, and this studied various interesting assemblies as a function of these parameters. You get nice phase diagram, and we could add details into this model as interaction varying across as a function of sequence and so on and so forth. And we studied this, where we had some interesting results. I just wanted to point out this. With this, I will conclude and stop. So the overall conclusion is the takeaway. What did we learn from this study? So the one takeaway, the first part is that the three-dimensional organization of the chromatin, the irregular structure can arise due to nucleosome plus non-histone proteins. So it is experimentally seen that irregular structures exist. One reason for this irregular structure could be the bending of bending due to the non-nucleosomal proteins. So if the density of non-nucleosomal protein is what is biologically sensible, one would get an irregular structure, is what we showed. In the microtubule disassembly regime, the bending elastic energy has to be comparable with the lateral energy. If the bending energy dominates, if that is much larger than the lateral interaction energy, it will unzipper very fast, is what our simulations show. And we also have a physical model for aggregation, where competition between elasticity and interactions, how do they lead to diverse assemblies? So these are this, and then finally I have an acknowledgement slide which is not showing up again. So these are the guys who did all the work. So Josna, who did the work on nucleosomes, Ishudesh, who's here, who did the work on chromatin, Gaurav, so these two guys graduated, and Gaurav, who's here, who did the chromatin 3D assembly, and Hungyo, he's also doing the chromatin 3D assembly work, who's about to graduate. And I have my colleagues, collaborators, Dibyendu, Samir for the protein aggregation, and John Marco, with whom I did a lot of nucleosome dynamics work. No, so we went for a wedding of one of our lab mates' sister's wedding. So that's the occasion we had.
Regular structure.
Yeah.
So what was the biological significance of that 50% or 30%?
Okay, so many of these proteins, so the concentration of these proteins are known. For example, the HMG protein, in the typical concentration known as, there is one HMG protein for every 2 nucleosomes. Like it's like 50% of nucleosome histones or this HMG. So that is, I do not want this number to be much larger than what is physiologically known concentration of these proteins.
So you're saying this 30 to 50% is what's biologically...
A sensible number.
Okay. And I had a second part, which is that in that model, in that same zigzag thing, do the nucleosomes slide or unbind? Or
no, they are fixed here.
Okay, but that could also be a source for destabilization of the zigzag?
Exactly, yeah, exactly, yeah.
Okay, but that is not low.
No, that's not accounted here. Yeah.
So my question is regarding chromatin organization, sir.
Yes.
So the histone tags, acetylation and methylation tags, does it have any role in organization of this chromatin?
Of course, they have important role. In a simple picture, one would imagine that acetylation, etc., would either stabilize or destabilize.
Okay, which further causes regulation in gene expression. That might be positive or negative.
What did you say?
That further helps in gene regulation.
Yes, so they help in assembly, gene regulation, and many things. Many of the modifications would destabilize histones, for example, or stabilize them, and so on and so forth. We have only assumed they all help to stabilize or destabilize this histone. So therefore, we have a stable histone because of certain modifications probably. That is why the histones are stable. Maybe one could argue that because of some modifications they are stable.
These non-histone proteins.
Yes.
So what would be the, what is the perfect structure that a chromatin should attain? Like you said, zigzag structure, instead of having zigzag structure, they're looking for some kind of regular structure by increasing the density of these proteins.
So what is known is not a regular structure. It's some kind of irregular structure. There is no perfect structure, at least not known.
Okay.
So people think.
So what are the side effects of this zigzag structure? Like, is it leading to any kind of unwanted gene expression?
We don't know. What is known is that it is not seen in experiment. However, in some cells like chicken erythrocyte cells where the known histone protein density is very small, in those cells one would see zigzag-like structure, which is also consistent with our prediction. If this non-nucleosomal proteins are very small, some cells have very small density of those, which they have this are seen. So why it is seen, not seen, I don't know, but this could be the reason why it is not seen, because there are other proteins, and if there are other proteins, of course, you would not have.
This zigzag structure is harmful to us, or
can we
don't
this
know.
Seems to be an extended discussion. Can we move on to the next question?
Yeah, so some of these HMG proteins do destabilize nucleosomes, which are not really included in your model.
Yes.
And they're found in regions that are highly transcribed.
Yeah.
And so they're beneficial, for like ribosomal regions, highly transcribed ribosomal regions. But what I also wanted to say is that we have done experiments with arrays of nucleosomes using AFM and adding NHP6A, for example.
Okay.
And when we reach a concentration that's near KD for binding the linker, we start to see stacking of nearest neighbor nucleosomes, which is basically exactly what's in your model. So it seems like there's some. We should talk more about that, obviously, but that's very interesting.
Yeah.
She had some questions.
Maybe I have missed. You have probably already said in your. So what you were showing that energy is required, ATPase activity is required for the structured arrangement of the nucleosomes. Yes. But in one sentence, if one has to say, what is the critical thing that is doing it?
Why is ATP required?
No. ATP is required, fine. But what is the key thing here, which is responsible for the structured arrangement?
Stability of nucleosome positioning, I would say, was one key thing.
But that is, I thought, the outcome of this. You want to show that these are the stable positions. But
oh, you mean why the stable positions? Okay.
How does energy?
Yes.
Because you're pumping in energy all the time and dissipating energy.
Yes.
So then what is the key thing that is giving rise to this ordered structure?
Okay, so there is, if you want to say one key thing, there is something called a barrier, which I didn't talk about today. That is supposedly a key thing near the transcription start site, and I will tell you about that after this. There is something called a barrier which would like a a block and which would do something, which I can tell you after that, which I didn't discuss today. That is supposedly a key thing for nucleosome organization near the transcription start site.